Lucky you! You saved 90 minutes of waiting in line because you have a sketchy biochem front of the line pass. It's finally your turn to participate in Sketchyland's biggest race ever at Pole Position Speedway. It's time to put the pedal to the metal. Remember to push the clutch all the way down and don't grind those gears. Will our defending champion Thunder Von King win again for the millionth time today? In this sketch, we'll talk about the pentose phosphate pathway. This race is brought to you by Cytosol, the makers of the best wiper fluid west of the Mississippi. The pentose phosphate pathway, or HMP shunt, takes place in the cytosol, specifically in mammary glands, liver, adrenal cortex, adipose tissue, and red blood cells. Oops, looks like we just missed the last race. Thunder Von King wins again. He's ribose 5-phosphate, represented by the ribose ribbon, number 5, and supercharged P battery. The pentose phosphate pathway produces ribose 5-phosphate, which is needed to synthesize nucleotides. The pentose phosphate pathway also produces NADPH, or, for the purposes of our sketch, petrol. We've used petrol instead of gas because petrol and NADPH both have the letter P. NADPH is an energy-carrying molecule that's needed for various processes, and petrol is the fuel that makes cars go vroom. This petrol-powered buttered popcorn machine will help you remember that NADPH is needed to make cholesterol and fatty acids. You'll see a guy filling petrol in a superoxide car. Check out that tailpipe burst. Fire in the hole! NADPH is used during respiratory or oxidative burst, which is critical for immune function and cell signaling. Macrophages, neutrophils, and monocytes use NADPH to produce superoxide free radicals from molecular oxygen via NADPH oxidase. These superoxide free radicals kill ingested bacteria and fungi in a process called respiratory burst. And I'm sure that burst probably killed any bugs hanging near that tailpipe. They don't just sell petrol here, they sell petrol with electrons, so you know you're getting top grade stuff. NADPH donates electrons to neutralize harmful reactive oxygen species. NADPH also regenerates glutathione in red blood cells. We'll use our universal symbol of glutes and thighs to show glutathione. While I applaud this gentleman for not missing leg day, let's backtrack. What's glutathione? Glutathione exists in the active reduced form or inactive oxidized form. In the active reduced form, glutathione neutralizes free radicals and peroxides to protect cells against oxidative damage. During periods of oxidative stress, the ratio of reduced glutathione to oxidized glutathione decreases. In other words, more reduced glutathione gets converted to the oxidized form. The enzyme glutathione reductase uses NADPH to turn oxidized glutathione back into the active reduced form. The mechanic holding a beer while working under the car will help you remember that NADPH is needed to metabolize large quantities of alcohol through the hepatic cytochrome P450 family of enzymes. If you don't remember cytochrome P450, think of these chrome fender flares. Finally, these heme hubcaps will help you remember that NADPH is required during heme metabolism. Alright, let's now switch gears to talk about each step in the pentose phosphate pathway. Before we begin, just know that we don't use or make ATP during the pathway. This ride has two parts, denoted by the different textures and colors on the roads. That's because the pentose phosphate pathway is divided into two phases, oxidative and non-oxidative. The oxidative phase comes first, and that one-way sign means that it's irreversible, meaning the reactions can only move in the forward direction. Glucose 6-phosphate, or G6P, the first intermediate in glycolysis, undergoes several irreversible oxidative reactions to become ribulose 5-phosphate during the oxidative phase. This route G6 produce truck in P battery is G6P. G6P dehydrogenase catalyzes the first rate-limiting step, converting G6P to 6-phosphogluconate, or 6PG. This worker dehydrating the water on the produce truck will represent G6P dehydrogenase. Note the petrol pump associated with the truck. That's because we make one NADPH during this step. Riders will encounter a runaway tow truck that fled the petrol station but crashed through the barrier onto the track. Let's listen to what the driver has to say.
I were just minding my business when this here guy done told me a car done broke down on the track and done need to be towed. So I done pushed the gas, but I crashed.
I think what he was trying to say is that the 6-hook and the pump gas will help you remember 6-PG. In the final step of the oxidative phase, 6-phosphogluconate dehydrogenase oxidizes 6-PG to ribulose 5-phosphate. Note the puddle of water and towel on the 6-PG tow truck. The tow truck ripped out a fuel pump when it fled. The hose is leaking petrol that caught on fire, producing black smoke. This step produces an additional NADPH and 1 carbon dioxide, which is represented by the petrol and black smoke. You'll see another car with the number 5 and P battery, but someone tagged "you lose." That's ribulose 5-phosphate. All right, we're officially done with the first part of the race, and now we're about to enter the second part, the non-oxidative phase. The two-way sign means that the non-oxidative phase is reversible. First, phosphopentose isomerase isomerizes ribulose 5-phosphate into ribose 5-phosphate. The guy switching 2 wires in the broken "you lose" car is phosphopentose isomerase. The next car looks just like Thunder Von King. That's because it is Thunder Von King, aka ribose 5-phosphate. Remember how we talked about how ribose 5-phosphate is used to synthesize nucleotides? Well, if you look at this pathway leading off the screen closely, you'll see nitrogenous bases on the pavement. Perhaps a shortcut? Maybe Thunder Von King should call himself Thunder Von Cheater. Sick burn. Anyways, let's direct your attention to these transport keys, which represent transketolase. Transketolase converts ribose 5-phosphate into glyceraldehyde 3-phosphate. Transketolase also produces fructose 6-phosphate. This questionable valet guy wearing a T1 shirt with 2 P-shaped guns is another thiaminator fan. If you don't know who thiaminator is and why he's important, please watch our vitamin B1 thiamine sketch. That shirt symbolizes thiamine pyrophosphate, or TPP, a cofactor for transketolase. We drew a truck with an ad for Glacier Ice to help you remember glyceraldehyde 3-phosphate, or G3P, which can enter glycolysis. Finally, this Fruit 66 fruit stand in P battery is our second product, fructose 6-phosphate, or F6P, which also enters glycolysis. All right, let's spend some time discussing G6PD deficiency, a very high-yield topic that you must know for exams. G6PD deficiency is an X-linked recessive metabolic disease that causes hemolytic anemia during periods of oxidative stress due to a decrease in NADPH. Red blood cells are susceptible to oxidative damage from harmful oxygen-free radicals and peroxides. Normally, red blood cells rely on glutathione to eliminate oxygen-free radicals. As we discussed earlier, G6PD is the rate-limiting step of the pentose phosphate pathway, serving as the predominant source for NADPH, which is needed to regenerate glutathione. The lack of available glutathione results in buildup of reactive oxygen species inside red blood cells and causes hemolysis. We drew broken watermelons to show hemolysis. G6PD deficiency causes hemolytic anemia during periods of oxidative stress. Patients are usually asymptomatic but develop symptoms like fevers, dizziness, fatigue, shortness of breath, back pain, and jaundice when they're exposed to infections, inflammatory conditions, fava beans, and certain drugs, including primaquine, aspirin, ibuprofen, sulfonamides, dapsone, and nitrofurantoin. These bacteria-shaped vegetables, pill-shaped vegetables, and fava beans will help you remember that infections, drugs, and fava beans cause hemolytic anemia in people with G6PD deficiency. Peripheral blood smear will show Heinz bodies, which form during periods of oxidative stress and represent denatured and precipitated globin chains. These watermelon seeds represent Heinz bodies. Splenic macrophages remove Heinz bodies to form bite cells, which are also visible on peripheral blood smear. And what better way to show bite cells than to take a bite out of a watermelon? Another important laboratory finding is hemoglobinuria. G6PD deficiency is more common in people of African, Middle Eastern, or Mediterranean descent, shown by this global delivery map. You should also know that G6PD deficiency protects against malaria. Parasites can't really grow inside red blood cells with low G6PD activity and high oxidative stress. We drew mosquito netting on top of the truck to illustrate this. All right, time to wrap up the sketch. Let's summarize. The pentose phosphate pathway takes place in the cytosol of mammary glands, liver, adrenal cortex, adipose tissue, and red blood cells. The pathway produces ribose 5-phosphate, which is needed to make nucleotides, and NADPH, which is needed to make cholesterol and fatty acids. NADPH is also required during respiratory burst, regenerating glutathione, metabolizing large quantities of alcohol, and metabolizing heme. The pentose phosphate pathway is divided into two phases, oxidative and non-oxidative. The oxidative phase is irreversible, and the non-oxidative phase is reversible. First, G6P dehydrogenase catalyzes the rate-limiting step of converting G6P into 6PG. This produces our first NADPH. Next, 6-phosphogluconate dehydrogenase converts 6PG into ribulose 5-phosphate. This produces our second and final NADPH, as well as carbon dioxide. Then, phosphopentose isomerase transforms ribulose 5-phosphate into ribose 5-phosphate. Finally, transketolase turns ribose 5-phosphate into G3P and also produces F6P, both of which enter glycolysis. G6PD deficiency is an X-linked recessive disease that causes hemolytic anemia during periods of oxidative stress due to a decrease in NADPH and low availability of active glutathione. G6PD deficiency is usually asymptomatic, but affected patients may present with fevers, dizziness, fatigue, shortness of breath, back pain, and jaundice when they're exposed to infections, inflammatory conditions, fava beans, and certain drugs. Peripheral blood smear will show Heinz bodies and bite cells. Hemoglobinuria will also be present. G6PD deficiency is more common in people of African, Middle Eastern, or Mediterranean descent and protects against malaria.